These are the directions for your regeneration lab. In this lab, you get to create new life using this planaria worm. The question we're investigating in this lab is, if we cut a planarian worm, how many days does it take to regenerate after injury? So this is something you guys will be able to measure and observe in this lab. So regeneration, this is the first thing you are going to write on your paper, is the ability to grow back or replace lost or damaged body parts. So this is not something that humans can do extensively, but this worm is totally capable of it. So when it is cut, even if you just have a little tiny piece of it, that piece will grow back into a whole new organism. So our first step is to learn more about this worm, this organism that you're working with. And so we have this section of your lab to write on. And so it's your job for the next few slides to listen to the information and record things on your paper. You pretty much have a choice of what to write here, and at least 3 things about the worm as we go over them. And then over in your diagram, you're going to be labeling. We'll go over that together, decide what kind of symmetry it has. And then some of these are more specific questions that will be answered, and it goes pretty much in order in the next few slides. So some facts about planarians. Their scientific name is Dugesia, and they are flatworms. They really are flat. You can kind of see that in the picture here. But as you watch them move and observe their behaviors, you'll notice that their bodies are flat. And if you look at the picture here, you get a little bit more information about their bodies. Each picture shows a different body system. So for a digestive system, their mouth and their anus are the same hole. It's called a pharynx. It's not really a hole, but more like a tube. So I'll let that sink in for a minute. For a nervous system, they have some eye spots, which aren't truly eyes, but it is something that helps them detect light in a very simple nervous system. They do not have pain receptors or similar sensations to what we would feel or experience, so it's not going to hurt them to cut them. So if you're worried about that, don't worry about that. We'll say more about the reproductive system in a bit, and yeah, more is coming. So characteristics of flatworms. They are the simplest kind of worm that's out there. Roundworms are even more complex than flatworms are. There's about 20,000 different species of flatworms, and they can live in many, many places on the planet. They can be free living, which means that they hunt down and catch their food just like a free living animal, or they can be parasitic, which means they have to feed on other living organisms. And another flatworm that does that is the tapeworm, which you might have heard about. Some people get tapeworms when they eat undercooked meat like pork. And most appear to have 16 chromosomes or 8 pairs. So you should be writing any 3 of these characteristics on your paper. So in terms of other body systems here, and again, you can write down any of these on your paper. There's no skeleton. It has tiny little bristles called cilia that help it move. It's got 2 layers of muscle under its skin. They do have what looks like a head region where the eye spots are, so you can figure that out. They sort of have a combination digestive excretory system. And we talked about this already. It takes food in and gets rid of waste through the same opening. Aren't you glad that humans don't do that? Nervous system, very simple nervous system, 2 nerve cords. It has 2 simple brains called ganglia, which are just basically bundles of nerves. They do not have a brain in the sense that we have a brain. They have a couple bundles of nerve cells that are sort of the coordination center, I guess, of the rest of their body. And then their eye spots that help them detect light. They have a gastrovascular cavity with 1 opening. So this is the circulatory system. It does not have a circulatory system contained like we do. It doesn't have blood on the inside of it like we do. Respiration, it does take in oxygen, but it has no formal respiratory system, no lungs, no gills, not like that. And then we'll talk more about reproduction in just a second. So here's a picture where you can kind of see all the systems one on top of the other. Here is the pharynx, which we talked about, which is the mouth and the anus. Here's the eye spots. You're going to label these on your paper in just a minute. So go ahead and label the eye spots. Hopefully you can figure out where those are on the body. Label the anterior, which is the head end, and the posterior, or the bottom end, on the diagram. So pause now and do that. Hopefully you notice we have 2 eye spots, and that this whole region is the head or anterior region, and this bottom part is the posterior region. We also wanted you to note the symmetry that the planarian has, and basically symmetry is just like what kind of body plan does it have. And the choices really are radial or bilateral. So things that have radial symmetry, no matter which way you sort of divide them in half, it looks the same on both sides. So for instance, this sea star has radial symmetry, but this butterfly has bilateral symmetry. This side is a mirror image of this side. If I cut it this way, it doesn't look the same, right? Because there's a head end and a tail end. So think about which kind of symmetry the planarian has. Hopefully you came up with the fact that it's bilateral, like us. It has a left and a right side. Asymmetrical, by the way, means there's no symmetry. You can cut it any way, and neither side is going to be a mirror image or the same. So where do these planarians live? Well, they can be found in many parts of the world, generally in fresh water, so rivers, streams, ponds, lakes, ditches, mud puddles. They tend to live on the underside of rocks, leaves, and other objects in very shallow water, as well as around or in aquatic vegetation like elodea and algae. So into reproduction, planarians are hermaphrodites. You may have heard this word before. They basically have both girl parts and boy parts. So if you look at this diagram again, they have ovaries near their heads. You can't really see them on the microscope very well. And then they have testes lower down in their body that produce sperm. So basically, they can reproduce both sexually and asexually. There's 2 methods of asexual reproduction. Fragmentation just basically is where they split apart and the pieces regenerate. And spontaneous dropping tails, which is sort of like spontaneous cutting themselves in half. So basically, they sort of, I don't know, stick themselves on something and pull apart, stretch their bodies apart, which is enough to kind of break their bodies in half. The head ends up growing a new tail, the tail grows a new head. And a lot of times this is happening in stagnant water, places where the conditions for living aren't great. So it kind of will stay like that until water conditions improve. And sometimes we've seen that happen, that dropping tails happen in, the cultures we purchase, like where there's enough food or space or, stagnant water that they're sitting in. So in sexual reproduction, each planaria gives and receives sperm, so they can reproduce with themselves, with their own gametes, or mate with another worm. But it is desirable to try to mate with another worm because you are altering the genetic code, right? Every time you mix DNA from 2 organisms, you're getting something unique at the end of that, and that could potentially give them an advantage. So here you can kind of just compare the 2 processes, right? So we are basically going to cause the planaria to perform asexual reproduction by cutting it, but they could swap sex cells and develop eggs or fertilized eggs, which would grow into genetically different planaria, genetically different from the 2 parents. So I want you to stop right now and make a hypothesis. I think that it will take how many days for the planaria to regenerate and give me a because? Do that right now. Materials. You're going to need a planaria, and it's going to be contained inside a petri dish, and these you'll pick up off of my desk. You'll need a dissecting scope, which I'll say more about in a minute. The scalpels you'll just need for today, and you'll find those at the stations, a ruler, a glass slide, and a dropper. So in terms of holding and handling the Petri dishes have two parts. They have a bottom dish that tends to be deeper than the top lid. Okay, so make sure that when you're handling your Petri dish, it needs to be correctly oriented. So the bottom goes on the bottom and the top goes on the top. What happens is if you have them upside down, it's like holding the dish this way, and the worm can end up swimming out of your dish and crawling up and out of your dish. So please make sure that you don't orient them that way. We also see that those dishes spill or slosh water around because the lid, where in this case all the water is sitting, isn't deep enough like the bottom of the dishes. So just be aware of that, please. You're going to be writing your names on the top lid of your Petri dish with a Sharpie. The stereo microscopes, a lot of you have noticed, are different than microscopes you've used. You actually have used these before back when we did Microscope Mania. They're just like a very large magnifying glass. So I know there's two eyepieces. It's a binocular scope, but really the power is about 20x, so 20 times what we can see, which is, you know, far less than our other microscopes that we've been using, but they are perfect for observing the kind of organism you're going to observe today. You have the ability to turn on two lights. There's a switch that shines light from the bottom and a switch that will shine light from the top. So you can turn on one or both of those, do what suits, you know, what you're looking at. And you still use the focus knob on the side to focus. This part moves up and down, and you'll notice it will go in and out of focus with turning that. So the basic guidelines for the lab are that you work in groups of 2 to 4, and I will let you choose who you're working with. A minimum of 2 people, a maximum of 4 people. Most people choose to be in groups of 2 or 3. However, each group member still has to complete their own paper. So your first step today would be to pick your group members. You'll then go back to my desk, pick up your planarian in the Petri dish. You're going to add your names to the lid with a Sharpie. And then you're going to follow the procedures on your paper, which I'll show you in just one second, collecting data as you go. And as we work our way through this lab, the care of your planaria is in your hands. So improper care and, for instance, loss of your planaria just from careless handling or not cleaning the water, that kind of thing, may result in you not being able to collect your data and may lower your grade. I would encourage you to share your data and compare what's happening with your planaria to others in your class and see, does everyone else get the same results you're getting? Probably that will vary on the type of cut you make, and we'll talk about that in a second. Okay, on day 1 of your lab, this is the information that you're going to collect, these 8 things. You're going to measure your planarian. You want to try to let it stretch out and measure it in millimeters with the ruler. Its activity level, does it seem to be passive or really active in the dish? Describe its movement. Does it swim freely or creep around? 4, describe where in the dish it spends most of its time. You're going to stream water at it. You're just going to push water at it with a dropper and describe how it reacts. You're going to be looking at these with a microscope, so you're going to look at its reaction to light and dark. So does it prefer the dark or does it prefer the light? And then eating behavior, this is something we'll do whole group and observe under the camera. So I don't know exactly what day we'll do this, probably day 1. But that's where you make that observation. And then finally, you're going to cut your planaria and describe that. So for this, there's detailed directions on your paper, but preferably what you're going to do is use the dropper to get the planaria onto your slide so that it's not in very much water, and wait for it to stretch out, and you're going to use a scalpel blade to cut it. So you have a decision to make about how or where you want to cut it. This one and this one, these two are the hardest cuts to make, and rarely have I had a class or person be successful in cutting them that way. This one, is probably the easiest cut to make, and this one, so bisecting or trisecting it. So choose that, describe your cut, and then I want you to look for and describe the movement of the planarian after the cut. At that point, then you will replace your planaria back into its Petri dish, put it back in the designated spot for the class, And then we'll be looking at the planarian over the next week or so. If you need to add water to your dish, you have to use the spring water, not the tap water that's from the sink. So spring water beakers are around the room. So as we work our way through the next week and beyond, you guys will be filling out the third page where you're going to keep track of the date, the sketch, and the observations, and I will go over that with you on Monday when we look at our first observation. So again, just to recap the day, today you will have made all your initial observations of the planaria. You will have used the scalpel to cut the planarian. You made observations after you've cut. Your planarian's back in the Petri dish, labeled. It's placed in the designated area, and your paper goes in a safe spot in your binder. And most importantly, this is your scoring rubric that shows how you're going to earn points. You get points for research, points for all your observations and data, 8 points for the initial observations you make on day 1, and another 8 points for the ones you make over the following couple weeks. You have a choice about what days you do that. And then you do the analysis questions that are at your last page and write a conclusion to our question. And then, of course, you also get some behavior points. So this all together adds up to 35 points. Please don't lose this paper.